Hello students. In today's class, we are going to learn about cell as a basic unit of life and its evolution. So what is a cell? Cell is the basic structural and functional unit of all living organisms. It is the smallest unit of life that can replicate independently. Cells were first discovered by Robert Hooke in 1665, and the discovery of cells was made possible through the invention of the microscope. He observed the cells in the thin slices of the bottle cork. Hooke discovered many tiny pores that he named as cells, and a name was derived from a Latin word named cella. He described that the cells appeared as tiny boxes or had a honeycomb-like appearance, and at that time he thought that cells existed only in plants and fungi. So here is the image of the Robert Hooke's microscope, the first uh, discovered light microscope. And here are the drawings of the structure of the cells that Robert Hooke observed under the light microscope, which had the honeycomb-like appearance. Uh, Anton van Leeuwenhoek in 1673 made use of a handmade microscope to observe the pond scum, and he observed single cell organisms which he named as animalcules. He also observed several blood cells from fish, frogs, dogs, humans, etc. Now, between Hooke and Leeuwenhoek discoveries and the mid 19th century, very little advancements were made in the structure of the cells, and this was probably because of the widely accepted belief that the cells have generated by the spontaneous method. So there were several examples at that time which suggested that the origin of life has taken place from spontaneous generation. For example, when dirty clothes were kept in a room, mice came out of them after a few days. And similarly, rotten meat was kept in a room, and maggots were developed out of these in few days. So without considering the fact that possibly the eggs of the maggots were laid on the rotting meat, and these eggs germinated later on to give rise to maggots. So at that time, the accepted theory was spontaneous generation theory. So people actually thought that if a rotting meat is kept for long, then it can give rise to maggots. Later on in the 19th century, many doubted the spontaneous generation theory, and it was disproved by Louis Pasteur. With this, there was development of the cell theory, which was based on findings of several scientists. In 1838, a German botanist, that is Sheldon, concluded that all plant parts are made up of cells. Similarly, a German physiologist, Schwann, in 1839, also observed that animal tissues are also composed of cells. A German physician, Virchow, in 1858, he extensively studied cellular pathology, and he concluded that cells must arise from pre-existing cells. Based on all these findings, the initial cell theory was proposed, which stated that all organisms are composed of one or more cells. Cell is the basic unit of life in all living things, and all cells are produced by the division of pre-existing cells. Later on, additions were done to the previous cell theory, and that gave rise to the modern cell theory. So the modern cell theory also took into consideration other factors or contributing factors such as the hereditary material or DNA. So based on the modern cell theory, which has four rules, the cell contains the hereditary information, that is DNA, which is passed on from cell to cell during cell division. All the cells are basically the same in chemical composition and metabolic activity. All basic chemical and physiological functions are carried out inside the cell. There's movement and digestion, and the cellular activity is dependent on the subcellular structures and their activities within these structures, such as nucleus, plasma membrane, etc. Broadly, cells can be categorized into two types, that is a prokaryotic and a eukaryotic cell. Eukaryotic cell contains a well-defined nucleus, while prokaryotes do not. Prokaryotes are also single-cell organisms, while eukaryotes can be either single-celled or multicellular. The prokaryotes were the first single-cell organism that lacked a membrane-bound nucleus, mitochondria, or any other organelles, and the cell division in them mainly occurred by binary fission. Example would be a bacteria. Prokaryotic cells were the first life form on the earth, and they are simpler and smaller than the eukaryotic cells. So here is a diagrammatic representation of a typical prokaryotic cell. So a bacterial cell or a prokaryotic cell can have varied shapes, such as it can be a rod-shaped bacteria. So here is an example of a rod-shaped bacteria. Looking at the structure of the bacteria in the cell cytosol, you can see that there is no well-defined nucleus present, but only the DNA or the genetic material is present, which is also known as a nucleoid. Then we also can see the extra chromosomal DNA in the form of plasmids present in the cytosol, and small organelles such as ribosomes, which are involved in protein synthesis. Here we do not have other organelles or compartmentalization of different organelles present in a prokaryotic cell. Coming to the membranes of the prokaryotic cells, we have an outer membrane and an inner membrane present. The space between the two membranes is known as a periplasmic space, and this space is deposited by a cell wall material which is made up of peptidoglycan. The function of cell wall is to give rigidity and a definite shape to the bacterial cell. There are different shapes to a bacterial cell such as foci, bacilli, spirali, vibrio, and so on. So this particular shape of the bacterial cells is because of the deposition of the cell wall material. We can also see an outermost layer of capsule that is present only in some bacteria. Not all bacteria can synthesize capsule, and it is basically a polysaccharide material that is secreted by the bacterial cells and is present on the outside of the bacterial cell covering the outer membrane. So the main function of the capsule is to basically hide the antigens that are present on the surface of the bacterial cell, so that they can help prevent phagocytosis of the cell. We can also see a flagellum structure that is present, that is used for the swimming motility of the bacteria. Apart from that, we can see some short hair-like structures extending from the bacterial cell surface, which are known as villi. There are two major types of villi. One are those which are involved specially in the conjugation purpose. These villi are known as conjugative villi. The conjugative villi allows for exchange of genetic material between different strains of bacteria. The other type of villi, which you can see, they majorly help in adhesion or attachment function to different substrates, such as it can be a host cell, or it can be also non-living materials such as stone or any surface. So this is about the basic structure of a prokaryotic cell. Next, coming to the structure of a eukaryotic cell. Eukaryotes have specialized organelles in the cytoplasm, such as it has a well-defined nucleus, it has a rough endoplasmic reticulum, Golgi apparatus, smooth endoplasmic reticulum, chloroplast, mitochondria, ribosomes, and other organelles such as ribosomes, lysozymes, etc. Lysosomes, etc. The size of a eukaryotic cell is much larger compared to that of a typical prokaryotic cell. And the cell division in case of eukaryotes occurs by mitosis or meiosis. Examples of eukaryotic cell includes plants, animals, fungi, protozoa, and algae. Next, we will discuss about the comparative properties of prokaryotic and eukaryotic cells. Example of a prokaryotic cell includes bacteria. That of eukaryotes includes algae, fungi, protozoa, and animal cells. Prokaryotic cells are typically smaller in size, which have 1 by 2 to 1 by 4 micrometers in size, while that of a eukaryotic cells are more than 5 micrometers in diameter. The genetic system consists of nucleoid, chromatin body, or nuclear material in case of a prokaryotic cell, while we have well-defined nucleus, mitochondria, and chloroplast present in case of a eukaryotic cell. There is no clear nuclear membrane and presence of a single chromosome in case of a prokaryotic cell, but there is a definite nuclear membrane bounding the chromosome or the chromatids in case of a eukaryotic cell. The chromosome does not contain histones or no mitotic division takes place in prokaryotes, while mitotic and meiotic division takes place in case of eukaryotes. Nucleolus is absent in prokaryotes, which is present in case of eukaryotes. Well-defined organelles such as mitochondria, chloroplast, Golgi body, endoplasmic reticulum, membrane-bound vacuoles are absent in prokaryotes while they are present in case of eukaryotes. Ribosomes are 70S ribosomes in prokaryotes while 80S ribosome in case of eukaryotes. Cell wall material made up of murin is present in case of prokaryotes, which is also known as peptidoglycan, while peptidoglycan is absent in case of eukaryotes. Pseudopodia are absent in case of prokaryotes, while they are present in case of some eukaryotes. Next, we are going to discuss about the endosymbiotic theory for origin of eukaryotes. According to this theory, the origin of eukaryotes has taken place by a endosymbiotic theory, according to which eukaryotes arose as a result of a fusion of an archaeal cell with that of a bacterium, wherein the ancient archaeal cell basically engulfed an aerobic bacterial cell. However, it did not digest it. The engulfed bacterial cell remained within the archaeal cell, in which later on a mutualistic relationship developed where the engulfed bacterium carried out the process of aerobic respiration by using oxygen to release stored nutrients, to release energy from the stored nutrients, and the host cell protected the bacterial cell from the predators. Over many generations, a symbiotic relationship developed between the two so that none of them would be able to survive independently. The descendants of these ancient engulfed cells are present in all eukaryotic cells today as a mitochondria organelle. Similarly, by engulfment of a photosynthetic bacterium and later on dependence of these bacterial cells and the host cell, development of chloroplast structures could have taken place in the modern photosynthetic eukaryotic cells. The same points are written over here for your reference. Now coming to the evidence for the endosymbiotic theory for the origin of mitochondria and chloroplast. First of all, they have the same size as that of the prokaryotic cell. They have their own DNA, and their DNA is organized in a circular chromosome similar to that of a prokaryotic genome, and their genome contains genes that are very similar to that of the genes that are found in the prokaryotic genome. Also, reproduction in these organelles takes place by binary fission, while the eukaryotic cells they will reproduce by mitosis. Finally, the eukaryotic nuclear genome do not control any membrane composition of the mitochondrial and the chloroplast organelles. So this actually suggests the origin of mitochondria and chloroplast in eukaryotic cells might have taken place as a endosymbiotic theory. So that's it about the first topic. Thank you very much, and kindly like, share and subscribe if you like the content.